In this video, we're going to talk about how the visual system processes color. What we perceive as different colors or hues are really different wavelengths of light. This process begins in the retina, and the photoreceptors in the retina that are responsive to color are called cones. There are three types of cones in the retina, and each of them is responsive to a different wavelength of light. These wavelengths correspond to what we perceive as blue, green, and red. According to the trichromatic theory, because we have these three different cones, each sensitive to a different hue, we can then perceive any other color by mixing those three lights together. These photoreceptor cones communicate with bipolar cells who communicate with ganglion cells. The ganglion cells of the retina responsible for perceiving color are parvocellular. Unlike the trichromatic process, the ganglion cells use an opponent process system with a center and surround that are antagonistic. To see how this works, stare at the small white dot in the middle of this flag. If you stare at this white screen for a minute now, maybe blink a few times, you should see what looks like a normal American flag. But how does this color mixing work between the photoreceptors and the ganglion cells? If we only have red, green, and blue cones, how do we ever perceive colors like yellow? Well, red is easy. Red cones are excitatory on red-green ganglion cells. So if your brain perceives an excited red-green ganglion cell, it knows you saw red. And green cones are inhibitory on red-green ganglion cells. So if the red-green ganglion cell is inhibited, then you perceive that as having seen green. Now, if you look over to the blue cone, blue is inhibitory on yellow-green ganglion cells. So that's easy. If a yellow-green ganglion cell is inhibited, you perceive having seen blue. Because yellow light excites both red and green cones, the net result of excitatory and inhibitory inputs leads to excitation of the yellow-blue ganglion cell, which you perceive as seeing yellow. Information from the retina travels through the optic nerve, the partial decussation at the optic chiasm, to the optic tract, where it synapses in the lateral geniculate nucleus of the thalamus. In this case, layers 3 through 6 are parvocellular. Moving from lateral geniculate nucleus of thalamus to the striate cortex, or area V1, primary visual cortex. The part of area V1 that's sensitive to color are the cytochrome oxidase blobs. Here you can see parvocellular information coming into 4C beta of striate cortex, and you can see the cytochrome oxidase blobs embedded within the tissue. As we move from area V1, the blobs send information through the thin stripes of extrastriate cortex V2, and then to area V4 of extrastriate cortex. If area V4 is damaged, one can develop a condition called achromatopsia, in which no colors can be perceived. And unlike a beautiful black and white movie, people with achromatopsia report everything looking like cement.